Yeah, welcome everyone. In today's discussion in the plant physiology and metabolism course, we will explore the intricate role of lipids. Lipids are a diverse group of organic molecules that are insoluble in water but soluble in nonpolar solvents. They play a crucial roles in various biological processes including energy storage, cell membrane structure, as well as that of the signaling. Let's start with the introduction of the lipids. The term lipid, coined by the German biochemist Bloor in 1943, is derived from the Greek word lipos, meaning fat. Lipids constitute a diverse group of compounds that includes fatty acids, fats, oils, waxes, and related substances. When lipids undergo hydrolysis, essential fatty acids are produced, playing a critical role in the functions of living organisms. Unlike carbohydrates and proteins, lipids do not form polymers, indicating a distinct structural characteristics. Notably, lipids exhibit insolubility in water but demonstrate solubility in organic solvent like benzene, ether, chloroform, and acetone due to their hydrophobic nature. These biomolecules, including lipids, are integral to the structure and functions of the cells, contributing significantly to various cellular processes and providing essential support. Biological lipids form a chemically diverse group of compounds united by their shared characteristics of being insoluble in water. Fats and oils serve as primary energy reservoirs in numerous organisms. Additionally, phospholipids and sterols play a pivotal role as major structural components within the biological membranes. Despite being present in smaller quantities, other lipids have a critical functions serving as enzyme cofactors, electrons carriers, light absorbing pigments, hydrophobic anchors for proteins, facilitators for membrane protein folding, that is chaperones, emulsifying agents in the digestive tract, hormones, and intracellular messengers. This diverse array of roles underscores the versatility of lipids in biological processes, making them essential components in both plant and animal systems. In plants, lipids are predominantly found in seeds and fruits, while in animals, they are present in adipose tissue, bone marrow, and nerve tissue. This distribution highlights the ubiquitous nature of lipids and their integral involvement in various physiological functions across the biological. So this slide shows the lipid metabolism. So the lipid undergo metabolism to generate a soluble form of sugars in storage organs, which are then transported to the growing regions in developing seedlings. While there are similarities in lipid metabolism between plants and animal, there exist significant differences. In plants, lipid metabolism is notably more intricate and involves various cellular organelles as shown in the figure, such as that mitochondria, glyoxysome, plastids, endoplasmic reticulum, etc. Fatty acid synthesis and oxidation occur in distinct subcellular locations utilizing different metabolic pathways and sets of enzymes. The oxidation of fatty acids involves the successive removal of two carbon units as acetyl-CoA. Concurrently, during fatty acid synthesis, the hydrocarbon chain undergoes elongation with the addition of two carbons at a time. Notably, the precursors for fatty acid synthesis are three carbon compounds, primarily malonyl CoA with one acetyl CoA acting as a primer. The condensation reaction during fatty acid synthesis is coupled with the simultaneous release of one carbon in the form of carbon dioxide. This intricacy underscores the sophistication of lipid metabolism in plants where diverse processes and subcellular locations contribute to the overall synthesis and utilization of lipids. This slide shows what are the role of lipids. Lipids in the form of neutral fats are commonly stored in the seeds of numerous plants as well as in fruits and other vegetative parts. Beyond their role as energy reserves, lipids serve as essential components of cell membrane, playing a pivotal role in determining membrane structure and organization. The amphipathic nature of membrane lipids, featuring both hydrophilic and hydrophobic ends, is critical for their arrangement within cell membranes. These lipids organize into bilayers with hydrophilic ends facing the aqueous environment and hydrophobic ends positioned inward. Membrane organization is driven by hydrophobic interactions between lipid ends and hydrophilic interactions with polar groups in the surrounding aqueous environment. The resulting biomolecular layer, in conjunction with proteins, upholds the integrity of membrane structure, a fundamental aspect crucial for life existence. The membrane, with its lipid protein composition, plays a pivotal role in creating and maintaining the optimal chemical environment within the cell and its compartments. By regulating limited permeability, membranes selectively control the entry and exit of molecules, thereby facilitating various metabolic reactions. This dynamic interplay between lipids and proteins in membrane underscores their significance in cellular function and the broader framework of life processes. Lipids play a diverse and crucial roles in biological processes. They serve as cofactors for numerous enzymes, including lipoic acid, ubiquinone, and plastoquinone. Essential fat-soluble vitamins such as D, A, E, and K are derived from 5-carbon isoprene units. Hormones, integral for both animal and plant systems, often originate from sterols. In plants, steroid, that is, steroid-like growth regulators, and jasmonates derived from linolenic acid contribute to various physiological responses. Lipids like cutin function as protective barriers in the epidermal cells of leaves, reducing water loss through a impermeability to water. Suberin, another lipid type found in the endodermis of root cells, acts as a barrier restricting the apoplastic movement of water from the cortex to vascular tissues. In plasma membranes, lipids also serve as signaling molecules, playing a crucial role in cell signaling. This multifaceted involvement of lipids underscores their significance in the structural, metabolic, and signaling aspects of cellular and organismal functions. As you can see here in the figure, the sources of the lipid molecules in the plant, that might be a triacylglycerol, phospholipid, a sphingolipid, brasinolide, as well as that of the methyl jasmonate. In plant, brasinolide is, that is a one of the active member of the brassinosteroids, are the growth regulator, which are steroid in nature. The methyl jasmonate, which is also a part of the jasmonate group, are derived from linolenic acid, and the jasmonate are also act as a plant hormone which regulates plant development, besides being involved in plant defense against insect, herbivores, animals, and many fungal plant pathogens. Lipids play a diverse role in biological system, as it has already been depicted in the whole table, and their functions are closely tied to their unique chemical structure. The triglyceride serving as a storage function, or esters of glycerol, where all three hydroxyl groups are esterified with fatty acids. The next one is the waxes, that is notably found in the jojoba plant, functions in storage and act as a protective layers on plant surfaces, deterring dehydration and predators. The next one is the phospholipid, which is vital for membrane structure, are amphipathic molecules with the polar phosphate group esterified with other polar groups like choline, serine, ethanolamine, or inositol. Cardiolipins, that is present in the inner mitochondrial membrane, participate in energy metabolism and mitochondrial apoptosis. The next one is galactolipids. It is devoid of a phosphate group, serve as a membrane lipids in thylakoids with the third hydroxyl group of diacylglycerol esterified with galactose sugars. Then we have a sulfolipids that are found in the thylakoids, involved in the esterification of two fatty acids to glycerol and a sulfocoinosyl group. Then we have a sphingolipids, that is amphipathic lipids that may be implicated in programmed cell death, replace glycerol with an amino alcohol called sphingosine. Then the next one is the sterols, acting as a membrane lipids and growth regulators like breast have a fused ring structure. Notably, cholesterol is absent in plants while phytosterols are present. The next one is cutin, which forms a complex structure that prevents free diffusion of hydrophilic substances, protecting aerial plant organs from dehydration and predators. The next one is the suberin, that is similar to cutin but containing dicarboxylic fatty acids and phenolic compounds, impedes the flow of water and hydrophilic substances in plant parts not exposed to air. Then the next one is the oxylipins, which contributes to defense against herbivores, include jasmonates derived from linolenic acid, and the last one is inositol phosphate, which is involved in the signaling of the cell, derived from phosphatidyl inositol phosphate. This comprehensive understanding of lipid functions and chemical nature highlights their critical roles in various biological processes and structural components across different organisms. So this slide shows the diversity of the lipid. A great diversity in the structure is observed in lipids. The quantitative catalog of all lipids present in a specific cell type under particular conditions is derived or referred to as the lipidome. The application of high resolution mass spectrometry techniques has proven instrumental in elucidating the lipidome of a cell. Notably, the lipidome of a cell exhibits dynamic changes under varying conditions, including differentiation, disease affliction, or drug treatments. The scientific field dedicated to the identification and comparison of lipids in different cell types under diverse conditions is known as lipidomics. This discipline utilizes advanced analytical tools to delve into the intricate world of lipids and understand their roles. Lipids can be categorized based on their structure or functions, and a fundamental classification distinguishes them as polar and nonpolar lipids. The exploration of the lipidome and lipidomics contributes significantly to unraveling the complexities of cellular processes and offers insight into the functional roles of lipids in health, disease, and various physiological processes. As you can see in the figure, which illustrates stearic acid. However, the figure B shows the oleic acid, whereas the figure C exhibits fully saturated fatty acid, and in the figure D depicts the mixture of saturated and unsaturated fatty acids. So now we have to stop here. In the coming lecture, we will divulge into the fatty acids fundamental role and other structure biosynthesis as well. So thank you very much for today's session.